PhD under the Department of Genetics and Plant Breeding at Navsari Agricultural University, Gujarat. So, first of all, what is plant breeding? So, plant breeding is the art, science, and technology to develop new variety which are better than existence variety according to market need using various molecular and genetic concept. So I am now talking about how to improve rice for salinity tolerance using novel tools and techniques. So you see in this picture, these two variety, this variety is tolerant to salt and this variety is susceptible to salt. You can clearly distinguish these two variety. This variety is very susceptible at the initial stage. So this is not efficient to reach up to the reproductive level, and this variety is tolerant. So this variety give more yield as compared to this variety. So this type of variety developed by using novel concept and techniques, and I will teach you how to develop these all type of variety. So rice is the staple food in all over the world, grown by 144 million farm family and 20 million ton, means out of 100% fertilizer, only 50% fertilizer used all over world in the rice. Yearly receive out of 100%, 35% of the water only consumed by rice. Annual revenue means $206 billion out of total crop value. And in export import also rice give more important. And 4 billion people all over the world out of 100%, 56% of the world population depend on rice. And see in this picture, rice provide more nutrient as compared to other, because rice is uh, as compared to other crop, rice is cheap. So poor people can also prefer and buy rice. So what is the health benefit of rice? So help to prevent cancer, prevent chronic consumption, cholesterol free, and control hypertension. Powder rice crush certain skin elements, great source of energy, vitamins, mineral, carbohydrate, reduce the risk of Alzheimer's and disease. Husk of rice means Lema Pela help the treat diastry, promote cardiovascular health, and give relief from irritated bowel the syndrome. So rice is the most important crop in our also India. So rice as a plant, this is the classification of rice, means how to rice uh, origin in our country. Common ancestors is Oreja species. So salinity is the major abiotic constraint limiting the rice productivity. It affects 800 million hectare of the land throughout the world. And see, in our India, maximum salt affected area in Gujarat, 2.22 million hectare, followed by Uttar Pradesh. It has been estimated 50% of the arable land will be saline due to salt accumulation. So, what is the major cause to increase salinity? First of all, low and erratic rainfall, increase evaporation, depletion of groundwater resources, and inappropriate irrigation practices, and also injudicious use of fertilizer. Farmer farmer think more, if we feed more fertilizer, then we we got more production, but it's not reality. High fertilizer is also affect and cause the day by day salinity problem. So uh, it affect after 1, 2, 3, 4 year and affect our next crop. Soil salinity along with drought is expected to be increased in the future. So development of salinity tolerant crop is most important because day by day population is also increased. So food food demand is also increased. So and soil is not increasing, but population is increased. So food requirement is also double. So it's developed to sustain rice productivity by using uh, develop salt tolerant variety. So rice is also very sensitive crop to salinity. Even at 2 EC lead to the 1 ton per hectare yield loss. And recorded the 50% of the yield loss when EC is 7.2. So excess salt cause both ionic toxicity and osmotic stress in rice plant. Under high salinity, rice plants show various morphological, physiological, biochemical alteration and symptoms and may die when the salt stress become very high. So this is the roadmap with the success of salt tolerance breeding. This is all over world cover means first of all 1969 beach land races. So Pokali, Nona Bokara, Chiruverupu, SR26B and G2. These are the salt tolerant land races. But day by day, linked drag day by day increase the how to develop salt tolerant variety using novel tools and technique means salt tolerance is physiological mechanism means this governed by genes and QTL. So without knowing any genes and QTL, we can't develop or introduce this gene into our cultivar. So knowing the genes and QTL which are govern this physiological parameter, identifying this physiological parameter and this parameter introduce in our local material. Suppose a Masuri, you all know Masuri is the very quality rice. So how to develop Masuri into salt tolerant? So this is Pokali is the salt tolerant donor. So in Pokali, one QTL. QTL means quantitative trait loci. This govern seedling stage salt tolerant. So this saltol locus introgates into the Masuri variety by using marker assisted backcross breeding, and then after this Masuri is also grow in the sea area means in those area where salinity is the major problem. But otherwise, normal Masuri can't grow in salt affected area, but develop Masuri with salt tolerance, it can grow in salt affected area, and it is most important for increase the livelihood of farmer which are living in the salt affected area. So uh, first QTL map Saltol by Chase et al. 1990. Then after this all means soma clonal variation use, anther culture, seedling stage, phenotyping. These are all the roadmap means day by day increase the knowledge and understanding about the salt tolerance and develop the salt tolerance variety. So rice, a glycophyte under saline condition. When rice, normal rice, not salt tolerant, normal rice, if we go in saline condition, then this is the morphological behaviors. If in seedling stage affect seed germination, means can't do germination, and at the initial level crop is damaged. Shoot and root length also reduce, shoot root sodium potassium ratio, seedling growth and vigor and chlorophyll content also reduce. And at reproductive stage, you see this is papery floret and chaffy panicle. Accumulation of sodium chlorine in the leaflet, high sodium potassium ratio, reduction in the photosynthetic active radiation, net photosynthesis rate and soma conducts. So photosynthesis, stomata, this is all reduced. And after this reduce, the productivity is very low. Means not able to produce any, say, per panicle only 5 to 6 grain is filled. all over panicle is disturbed. So this is the when salt increase, this is the effect cause in plant. See, this Navin is salt susceptible variety, and AC for 1585 is the tolerant variety. See, in susceptible variety, the symptoms like leaf rolling, spikelet sterility, degeneration. But see, in tolerant variety, this much is not affected. They complete its life cycle and see some output is give means the production is up to 4 ton or 5 ton per hectare. So morphological response are stunted plant growth, leaf burning, fluorosis, low tillering, leaf rolling, poor crop growth, effect on crop productivity like the spikelet sterility, less folate per panicle, less grain weight, low grain yield, low harvest index, and biological response means oxidative stress, alter the metabolism, high sodium transport to shoot. This sodium is more toxic to plant, means disturb the all metabolic process in plant. Uh, Low potassium uptake, and calcium and potassium is very helpful to reduce the sodium transport from root to shoot. Low phosphorus and zinc uptake, physiological response, inhibition of photosynthesis, stomata closure, decrease the water content, high amount of osmolyte and low osmotic acid. So how to fight rice under saline condition? So this side is tolerance and this side is susceptible. So how tolerance mechanism govern in rice to increase its or successful its life cycle. So high proliferation of root cap means sodium exclusion, means remove the sodium from its root side. Intracellular compartmentalization restrict the sodium into the root zone, and ion exclusion, intracellular compartmentalization sodium to vacuole, accumulation of various osmolytes. So it's maintain the turgor pressure. So these are the various breeding tool for developing salt tolerant variety. So screening of tolerant and sensitive germplasm. Without natural variability or germplasm, we can't start our precise breeding program. Variability is must before any starting of the breeding program. Germplasm include wild rice, some elite material, land races, modern variety, local variety, which are the source of genes which are govern the salt tolerance. Okay, so this all type of germplasm used in breeding program. These two type now conventional breeding program and modern breeding approaches. So first of all, conventional breeding program. First, introduction. Introduction means where the salt tolerant material is there, we take and introduce in our area. So this is introduction. Pedigree selection means suppose Masuri is the salt susceptible variety. So crossing Masuri with Pokali and their F2, F3, F4, F5, F6, we select the material according to our market need, but screening at salt affected area. When used backcross breeding, backcross breeding is the method when one or two trait we can transfer by using backcross. Recurrent selection means selection by using population improvement, means number of genotype or number of segregating material, those are salt tolerant, intermating, then generating new material, then after selection we can develop population. Mutation means sudden heritable change. So some mutation is beneficial for our life, like one of in next slide I will show by using mutation breeding we can develop salt tolerant variety. These are the technological innovations after 2000. Identification of QTLs, which are govern the salt tolerance, its fine mapping, genome-wide association study, teeling, eco-teeling, omics approach. These are all the tools which are help in tuning, fine tuning of the particular tolerance mechanism. Means which gene, which metabolite, which are responsible for uh, providing the salt tolerance. And by using this all the source gene, we can use in marker assisted breeding, transgenic approach, genetic engineering, and genomics. In my PhD program, I am using marker assisted backcross breeding to develop salt tolerant variety. Transgenic approach means any other organism, not like plant, any other organism which are having the salt tolerance mechanism catch up this gene and introduce in our material. Genetic engineering means expression, overexpression, down regulation of some genes which are responsible for salt tolerance. Genome editing means not alter the whole genome, only precisely alter one or two genes, and we can develop salt tolerant variety. So conventional breeding is not a proper way to develop salt tolerant. Why? Because salt tolerance is very complex mechanism and governed by a number of physiological parameters. So by without knowing any genes and metabolite, we can't select any trait which are governed for salt tolerance, because in conventional breeding, large environmental effect. So using modern breeding approach, we can easily and efficiently and uh, throughout running product in the market. And then this all approaches to after that evolution of improved line in a pot or field condition, and finally improved cultivar, which are salt tolerance, high yielding, improved trade, short life cycle. See, in a stress breeding, mostly variety are 90 or 100 days, not more than this, because short life cycle variety is helpful in stress condition. Okay. This is conventional breeding. So I am also telling salinity is complex trait, phenotypic accuracy is very low. These are all the traits which are measured during the salt tolerance. But without knowing any genes, we can't precisely interrogate and develop variety. So large environmental interaction, early segregation selection is not possible. Plant population is not sufficient and can't be weighed. That's why conventional breeding is not as a uh, helpful or precise to develop salt tolerant variety. Uh, but conventional breeding have bright side. These are the natural variability for salt tolerance, interspecific variability, gene pool 1, gene pool 2, 3, interspecific variability. In all over world, mostly as a donor parent, Pokali and Nona Bokara use for interrogation of salt. In my PhD research, this Nona Bokara is using IM as a donor parent to interrogate salt tolerance in our local material, Gujarat 70 variety Sardar. Pre-breeding material, these are FL470 is mostly used, and these are release variety. Pusa Basmati 1121 Saltol. This variety developed by Dr. Gopal Krishna and Dr. Sukumar Singh. This is the list of rice land races and release variety in India and all over world. These all varieties released in India for affected is area where salt is major problem. In Pakistan, Bangladesh, all these variety. I telling the mutation breeding is also very helpful to by using some gamma rays, X-ray, neutron, chemical mutagen to induce variation and capture useful variation which are responsible for salt tolerance. See, By using this breeding method, Suha 92, Basmati 370, NIAB Rice 1, TSR 1, 84-7. These are variety developed by salt tolerant mutation breeding. Chemical mutagen also use ethyl methyl sulfonate, and using this chemical mutagen, mutate one is developed. Another technique is tissue culture. In tissue culture, various techniques include first is anther culture. Means anther means male organ taking as a as a source, and we can develop variety by using double haploid technology. So double haploid technology is very useful because shorten the development of homozygous and uniform breeding line by 5 to 7 years compared with the conventional breeding. But anther culture depend on donor plant genotype, microspore developmental stage, cold treatment, induction, and differentiation media. So this is the criteria. Means when we use anther culture, these are must know, means which type of material we can use. So every genotype is not respond to tissue culture. Every genotype different response. Generally, indica variety is relatively low response to callus induction and regeneration efficiency. Using anther culture, ERI International Rice is a distributed Philip and developed salt tolerant rice PSBRC 50 bio. These are another varieties. Also, another variety is in India, CSR 21, CSR 28 is also used for cultivation in saline alkaline soil of India. This also two variety developed by double haploid breeding technology. So what is somaclonal variation? So variation in the callus culture means any tissue of the artificial growing media, and exploit this variation to develop salt tolerant variety. Is how to develop. So this mutant cell reach with the salt, and those cell is survive in salt condition. We can exploit this cell and grow in another culture media and develop whole plant. After that, we can also screen for salinity tolerance. So IR ERI Philippines improve the Pokali for because Pokali is salt tolerant, but having the unfavorable character like plant height is tall, so lodging problem, red pericarp, poor cooking quality. So improving of this Pokali for agronomic performance by subjected into in vitro induction of somaclonal variant and gate variant. This variant have improved agronomic performance coupled with good salt tolerance, having vigorous growth, semi-dwarf nature, white pericarp, and better cook quality rice. Another is Atbaki et al. Developed salt tolerant light from Malaysia rice cultivar MR263 via somatic embryogenesis. What is somatic embryogenesis? Means embryo of the seed extract, and by using this embryo, we can develop a whole plant and screen this plant for salinity tolerance. In vitro mutagenesis combine in vitro culture and mutagenesis through physical mutagenesis. Have been successfully utilized in the genetic variability and developing abiotic stress tolerant rice. So this is the screening, means how to screen our material for salinity tolerance. So this is a seedling float, means mostly rice is susceptible to major two stage. One is seedling stage and another is reproductive stage. So how to screen our seedlings for salinity tolerance, means which seedlings or which genotype is tolerant to salt and which genotype is susceptible to salt. By using this method, we can screen our material. uh, this work progress at this is my research, uh, my research photo. Actually progress at Navsari Agricultural University. So see this is my recessive parent and this donor parent. This donor parent is salt tolerance. So using this donor uh, genes, I am introduce this gene in our material. So see this is salt tolerant and this all are higher. 28 is also very susceptible to salt. Okay. So, which observation we can take during the screening of salt tolerance? So, plant survival, injury score, shoot length, root length, seedling fresh weight, shoot biomass, root biomass. Uh, these are evolution score, means highly tolerant, medium tolerant, susceptible, highly susceptible. Which are physiobiochemical? So, chlorophyll content, shoot sodium content, shoot so potassium, sodium potassium ratio in root and shoot. This is a screening technique at reproductive stage. So our material, it's not necessary that our material is resistance at seedling stage, and this material is also resistance or tolerance at reproductive stage. Seedling stage tolerance and reproductive stage tolerance both are governed by different genes and QTL. So introgation of these both two into single genotype is more appropriate. But in some area only problem at seedling stage. So scientists develop seedling stage tolerance. In some area the problem at reproductive stage. So scientists develop reproductive stage tolerance material. This is a flow diagram of how to screen our material at reproductive stage. And see, in this water, we continuously maintain the EC level at 10 or whichever salty problem area. Means in our area is 6, 7, 8 is the EC so problematic. So if we develop 10 EC sustain varieties, more useful for farmers. These are the trait under consideration at reproductive stage. This when we examine our plant, this all type of trait we take, and after taking this all trait we can we can examine which genotype is suitable for releasing a variety for salt tolerance. So in 21st century, uh, how to develop salt tolerance variety using multi-disciplinary approach, means advance in microbiology, advance in physiology, advance in breeding system, advancement of plant molecular biology. So nowadays is the day of molecular biology. So using marker assisted backcross breeding, genomics approach, phenomics approach, transcriptomics, proteomics, metabolomics, candidate gene, tealing, eco-tealing, association mapping, GWAS, QTL mapping, GBS, transgenic, soil metagenomics, functional genomic, genome editing, use RNAi, genomic selection. These are all the techniques which are now currently used scientists to develop salt tolerant variety. Means these all techniques are very useful to fine tuning of the tolerant genotype, means which type of chromosomal location which are responsible for salt tolerance, which physiological mechanism which are responsible for salt tolerance, identifying this character and precisely alteration of this parameter, means how to develop this how to importance of this physiological parameter to develop salt tolerant variety. So these all technique are very useful as compared to conventional breeding, because in conventional breeding without knowing we develop variety based on only phenotype, but phenotype is not accurate the performance of genotype, because environment is the major role. So using these novel techniques, identifying locus, QTL, genes, physiological parameter, metabolite, alteration of these parameter and develop salt tolerant variety. So what is the QTL mapping means? How to map particular genes which are responsible for uh, salt tolerance means develop, first of all by for QTL mapping we need mapping population like F2, DH, double haploid, REAL, NIL, chromosome substitution line, advanced intercross line. These are means suppose I am excellent. Suppose a real population. Okay, how to develop a real population? First of all, choose two parent which are extreme for tolerance. Means one parent is tightly or mostly susceptible to salt tolerance, and another parent is highly tolerant. So cross between these two parents develop F1, F2. From F2 to F5 or F6, we can use single seed descent method. Uh, and after up to F5 or F6, 400, 500 individual we can get. And this 400, 500 individual for test for screening salt tolerance at a reproductive stage and seedling stage, and taking the accurate phenotype. See, accurate phenotype is most important. Without accurate phenotyping, it's difficult. Means phenotype means result is mismatching. False result we can get. That's why phenotype is most important. So taking accurate phenotyping, genotype using 200, 300 molecular marker. Now SNP is the king of molecular marker, but SSR is also used. So these genotyping data and phenotyping data use for development for finding out which QTL is responsible for salinity tolerance. These are the various QTL mapping method like single marker analysis, simple interval mapping, composite interval mapping, multiple interval mapping. So, and these are the various software means map maker, cartograph, SAS. These are to phenotyping data and genotyping data put in this software, and software make our QTL means which QTL is responsible for salinity tolerance. This type of map we can get. See, this is chromosome 1 and this region is responsible for salinity tolerance at reproductive stage. These are also genes, but phenotypic variation is very low. This QTL is the phenotypic variation is 43%. They also called saltol. And in saltol, this saltol is present in Pokali. You are all know Pokali is the salt tolerant land races. And this saltol in between SKC, one is gene is responsible for also sodium potassium homeostasis in shoot, and also found in Nona Bokhara. Nona Bokhara is the West Bengal land races which are responsible for salinity tolerance. And chromosome number 8 and 10 is also having the QTLs for reproductive stage tolerance. See, chromosome 1, 3, 4, 6, 7, 2, and 9 having the maximum number of salt tolerance QTL reported. See, this is the saltol locus, which are responsible for in chromosome 1, responsible for salinity tolerance. Other than this, Benjamin et al. Reported some new QTLs, which are not present in Nona Pokhara and Pokali, using real population of Hasavi and IR29. Did not contain the same salinity tolerance level at Nona Bokhara and Pokali at SKC1 locus and Salto. So this Hasavi variety is salt tolerant and also having the other mechanism other than non-avocada and vocale. So these are the different trade which are correlated with salt responsive QTL. So see, iron homeostasis is the highly correlated with the salt tolerance in rice. Okay. So see, Hossein et al. Reported 16 QTL with 4 and 47% phenotypic variance in chromosome 1, 7, 8, and 10 in F2 mapping population developed from cross between reproductive stage tolerance from the donor Chiru Verupi and Pusa Basmati one. Okay. And this PSPS1 gene also play a key role in the salinity tolerance by maintaining photosynthetic activity. So these are the number of QTL and its function at which stage it's governs salinity tolerance. So this is the parental line which are used for developing or used for making the QTL for at the reproductive or saline stage tolerance. Another approach is association mapping. Mapping is two types. First of all, linkage-based mapping and one other is association mapping. So in association, suppose rice is easy to develop mapping population, but if we develop a mapping population in mango, then is the big question because mango life cycle is 10 years. So first 10 year is required to mango at reach reproductive stage. Another 10 year is for its F1 and another 10 year is for development of F2 book. So scientists waste his 30 year for only development of F2 population of mango. So what is the another uh, option to map salinity tolerance at perennial tree or another any other crop? So association mapping is most useful approach to mapping the genes and QTL. So its population-based survey used to identify trait marker relationship based on linkage disequilibrium by exploiting historical and evolutionary recombinants event. So these are the which are tools which tools and advantage over linkage mapping. Association mapping is population and jump plasm precise phenotyping, genotyping, structure and kinship analysis, LD analysis, association mapping and LD analysis. So this uh, field design data analysis. Okay, these are all the software which are used in association mapping. Uh, these are association mapping used for salinity tolerance in rice. During see chromosome 1 for 6 and 7, 44 significant SNP identified by Kumar et al. Most of the SNP for sodium potassium ratio were located in chromosome 1 near Saltol. So this Saltol locus is more helpful and efficient to interrogate and develop salinity tolerant variety. During reproductive stage, significant SNP increase under salt stress as compared to the unstressed condition. Mostly SNP sodium potassium ratio were detected in salt or region. 7 SNP associated with salt responsive QTL have been identified by Pimpong et al. 2014. So another technique is marker assisted selection. So when use marker assisted selection, when we know actual link gene or link marker, without link marker we can't start our breeding program or marker assisted selection. So DNA polymorphism is prerequisite for molecular breeding. Means at conventional level variation is most, but at DNA level, DNA polymorphism is required for starting molecular breeding. So see this, okay. So this is resistance variety and is susceptible variety. Okay. So resistance variety having the band is a small size, and susceptible variety have band is large size. Small size have run fast, and susceptible uh, big size band is move slowly. So crossing between those F2, and we select this type of band, this genotype, which genotype having this type of material, we can select this and develop resistant variety. So see, in this is resistant genotype. These two is also resistant genotype. This resistant genotype, and select this resistant genotype, and we can develop salinity tolerance. See, marker assisted. So marker assisted selection is very helpful. How? Reduce the population size. See, and this much of population we reject at the seedling level and only handle this population. So is time saving method, and also labor low labor requirement used in a marker assisted selection. So marker assisted breeding use molecular marker to reconstitute the desirable genetic makeup of the variety while introducing one or more important trait uh, during the varietal development in order to improve the efficiency of selection. So recently, Rana et al. 2019 transfer salinity tolerance gene from salt tolerant donor casein into high yielding variety Yukinomai through marker assisted selection coupled with speed breeding to develop salinity tolerant variety. So see this Pusa Basmati 1121, PB6, AS996, this all variety developed by using marker assisted selection to develop link marker assisted to develop salinity tolerant variety. See, this is the flow diagram of marker assisted backcross breeding, means how to select our plant which are actually resistant to salinity. So first of all, cross between this BR28 and Pokari develop F1. This F1 is through F1 identify and cross this F1. See, always F1 use as a male pair. Don't miss, because and a recurrent parent always use as a female pair. Why? Because if F1 we use as a female parent, then selection for through F1 and segregating material is missed. Okay, that's why female is always used as a recurrent parent, and F1 used as every segregating generation we use as a male parent. Okay, so crossing this F1 with recurrent parent BR28 develop BC1 F1 population. In BC1, try to keep a population at minimum of 400 or 500 population size, because this much of population give freedom to select our desirable type. Our selection efficiency is increased. If we select 100 plant type, so not this much of efficiency to select our desirable plant size. So keep population size at 200, not 200, 400 and 500 population size. In BC1 F1 population foreground and recombinant selection carried out. What is the foreground selection? Means selection of uh, selection of locus which are responsible for salinity tolerance, means saltol. So saltol at molecular level foreground means selection for particular trait, and in background selection of region which are similar to our recessive parent, means BR28. So we require BR28 variety with salinity tolerance. So genome recovery, how to increase genome recovery? By using foreground and background selection. Foreground selection for salinity tolerance and background selection for mostly genes which are responsive, mostly genes which are present in BR28. So after end after ending of this after end of this program, we can get BR28 same as normal BR28, but additional salinity tolerant, additionally salinity tolerance. Okay, so MAB scheme using focally as the saltol donor and BR20 as recipient line with number of genotype. See this. So BC1F1 foreground background selection. Select so 185 total plants. From this plant we can select 20 plants, and from these 20 plants we can select 3 plants which are similar to BR20 and develop BC2F1 population. Same, uh, same is BC1F1, BC3F1 selected. Same means foreground, background selection. Okay, and this line is same version of BR28 plus additional salinity tolerance. Okay, nowadays the era of next generation sequencing means low cost totally sequence the rice genome and which are genes and SNPs and all responsible for salinity tolerance identified by various techniques and genes. So nowadays sequencing is very low cost technology. So, so generally next generation sequencing facilitate the development of new tools such as MuteMap, QTL-seq, RNA-seq, and GBS. In MuteMap approach, the causal mutation can be rapidly identified by whole genome sequencing of pooled DNA from segregating population. So identify the loss of function mutation responsible for salinity tolerance in the Takagi 2015. What is the QTL-seq approach? So combine the power of BSA and NGS, assisted high throughput genotyping to identify QTL, controlling the complex traits such as salinity tolerance. And then what is the RNA-seq? Is identifying novel salinity tolerance gene by analyzing cultivar specific stress responsive transcript, means which RNA or protein product is responsible for salinity tolerance. Then next is phenomics and genomics. So what is the phenomics? So phenomics is a systematic genome-wide study of the phenotype of organisms. Germplasm evolution and varietal development through field evolution and novel phenotyping protocol for target cell stress factor have not taken to expected advantage of spiritual development in molecular and capitalism biology. And genomics is the interdisciplinary approach of field of biology focusing on structure, function, evolution, mapping, and editing of genome. The genomics and transgenic technology have so far provided little success in development success of salt tolerant rice variety for actual field condition. So accurate phenotyping and accurate genotyping is accurate because DNA is qualitative in nature, but phenotype is phenotype disturbed by more and more factors. So precise phenotyping is required to clarify the which trait are responsible for stress tolerance. So the difference between phenotyping and phenomics. The phenotype is the determination of type and level of expression of concentrate. As salt tolerance is complex and quantitative, it is challenging to task to carry out accurate data. So accurate phenotyping is far more difficult than genotyping because nowadays genotyping is very simple because the is the era of next generation sequencing. In a single day, we can sequencing all the genome of rice. So it's simple, but phenotyping is difficult. And it's the bottleneck for progress in basic plant science and plant breeding. So, how to take precise observation. So, using phenomics approach, concentrate with the measurement of phenomes, where phenome is the set of phenotype, physical and biochemical type. Integrative approach of plant biology, mathematics, engineering, and computer science. Uh,
Sir, sorry to interrupt. We have completed 50 minutes, sir. If you can quickly finish it, I'll take up the questions from the participants.
Hello, ma'am.
Yes, sir. Sorry to interrupt. We have completed 50 minutes, sir. If you
Uh,
can quickly finish it, I will take up the questions from the participants.
oh, okay,
Huh?
ma'am. Okay.
Okay.
Okay, then the phenotype is also required to develop salinity. These are we already known what is the genomic selection. I tell already in previous slide uh, which genomic selection how to use to develop salinity. So these are the all the wild rice which are govern the salinity tolerance. And so phenotype is also equally important for genotyping or genomic study. So which are tools required for phenomic studies? So magnetic resonance imaging, thermal imaging, chlorophyll and photosynthetic fluorescence, light detection and ranging, canopy spectral reflectance. These are all the computer-based tools which are used in a phenomic study and get accurate phenotyping data which are responsible for salinity tolerance. What is the transgenic approach means gene are taken from other species in order to get insertion of gene controlling the trait without disturbing any other desirable trait in recipient elite genotype. So, recently, salt-tolerant transgenic rice developed by using overexpression of gene OSIF from wild rice, which are native to India, Bangladesh, Sri Lanka, and Myanmar. These are the genetic engineering means overexpression or introduction of various genes to develop varieties. So, this list of genes which are used in Oryza sativa to improve salinity tolerance. See. These are all these genes which are responsible for salinity tolerance. Another is genome editing. So genome editing include homonizing endonuclease, zinc finger nuclease, talase, and CRISPR-Cas9. Time is very low, so I am forward fastly. If any question, then these are genome editing technique means how to edit rice genome, develop a stress tolerant variety. See, this is step. Okay. Other approach like eco-tailing, allele mining, whole genome resequencing, transcriptome. Okay. And another is Ocean agriculture means. However, despite all this progress, hope rests on the CRISPR-based effort of Agricia who intended to grow rice in the ocean practices called ocean agriculture and to overcome seawater tolerance barrier. The company's approach to producing genetically engineered salt-tolerant rice is based on editing network of genes, having edited 8 differently genes and on rice which on the plant is naturally adapted to high salinity condition. This floating ocean rice farm are expected nearly in the end of 2021. Another one land disease Oryza corcata. They grow in salty seawater area. So corcata is a very salt tolerant genes. These are the conclusion and future prospect. Uh, okay, it's no. Thank you. See, these are the salt tolerant variety and the salt susceptible variety. Simply interrogates the gene From these both variety are same, but this variety is developed by using or introduce some salinity tolerant genes, and say this is the normal variety C, and this is also salt tolerant and this salt susceptible variety. Okay, thank you, madam.
Thank you very much, sir. Thank you for a detailed presentation.
Mm.
I'll take up the questions from the participants now. In the meantime, can you please stop sharing the screen, sir? So that
Ma'am.
we can see. Please stop sharing the screen, sir, so that we can see your full video.
Okay, ma'am. Okay. Okay. Mm.
Okay.
Okay. Mm.
I'll move on to the questions.
Mm.
The first question is, why salt tolerance in rice crop is so important for farmers?
Okay, ma'am. Day by day, uh, the erratic rainfall, farmer use in judicious use of fertilizer and other practices means groundwater depletion. So this type of practice increase the salty problem in agriculture field. Means oh, crop, suppose rice is the very sensitive crop. Means salinity cause the rice production at 50%. If rice give 5 ton per hectare yield, but if salinity is the problem, then rice give 1 ton or 2 ton maximum. So sustain the production of various crops, we require to develop salinity tolerant variety. And see, in our Gujarat, mostly day by day sea water is increased. So it's need to sustain the productivity of rice, because day by day population is increased, but soil is not increasing. That's why salinity tolerance is important for particularly in rice.
Okay. Sir, can you please tell us about yourself? What made you take an interest in agriculture and opt for a research career?
Uh, okay, ma'am, I am basically from a farmer family. I completed my BSc at College of Agriculture, Vagai, MSc in the Navsari Agricultural University in the major research topic of F2 variability analysis for rice, and also in a PhD I am taking because I particularly interest in rice crops, and taking in PhD my research now marker assisted backcross breeding to improve salinity in rice. So I like rice, and from a BSc I am clear with the take of this subject, because genetics and plant breeding is uh, uh, genetics and plant breeding is mostly useful for developing new variety which are benefits for farmer, and farmer get a smile from this our better variety. So it's very huge and uh, uh, important social activity to uh, improve the life cycle of farmers. Hello, ma'am.
Okay, thank you very much, sir. We have now completed the round of questions. On behalf of agricultureinformation.com, we like to thank Mr. Prajapati Maulik Kumar Rameshbhai for the talk and for answering the questions. I like to also thank all the participants in this meeting. This meeting will now be closed.
Okay, okay, thank you, ma'am. Thank you.
You're welcome, sir.
Okay.
Thank you, Maulik.
Chetan,
Sir,
Chetan.
sir, I think he's from your side.
Yes, yes, yes. This is
If Mr. Chetan is having any questions, he can ask, sir.
No, no,
Chetan.
ma'am. No
Okay.
question.
Okay, thank you
Ma'am,
very much.
Okay,
Chetan,
ma'am.
ma'am. Hello,
Yeah,
ma'am.
tell me, tell me.
Chetan is my best friend. This is my MSc classmate. He is also working in Sayaji seeds,
Okay.
basically in cucumber and mustard breeding.
Okay, very nice. Maybe we will invite him also one day for
Yes.
the meeting. Okay.
Okay.
Thank you very much, sir. Okay.
Okay.
Thank you,
Thank you.
ma'am. Thank you, Maulik.
Thank
Okay.
you.
Thank you, Chetan. Thank you.